Hello, my name is Björn Ommer from University of Munich, where I'm heading the Machine Vision Learning Group. Today, I'd like to present a machine learning approach for behavior analysis that trains without supervision and provides us with a visual microscope for videos that learns to directly magnify in the video data in subtle abnormalities from reference behavior. It is clear that changes in cortical wiring due to disease or medical treatment can have negative or positive implications on motor behavior. Can we turn this around? Can we have an AI that just by watching changes in motor behavior of an animal or human patient can make meaningful predictions about the underlying neuroplasticity? This allows to watch a subject over multiple sessions and quantify the degree of neurodegeneration or compare the impact of different treatment paradigms on the behavior. So we want to automatically localize motor impairment, quantify the deviations, and visually magnify deviations from a reference directly in the video to give clinicians or scientists an accessible tool for actually seeing how a subject deviates from reference behavior. This comparison should work across different subjects despite large deviations in appearance, and all of this should be learned without requiring tedious manual annotation. Now, the state of the art in motor function analysis either utilizes physical markers, mocap, or similar extrinsic marking, which essentially distracts rodents and which is also very time consuming. Due to their size, such physical markers are also not well suited for analyzing the small paws of mice or rats. An alternative is visual key point tracking, such as the classical tracking by recognition approaches and more recent renderings, such as the very popular deeper cut and deep lab cut approaches. These are great tools, but they entail their very own limitations. For instance, doctors need to know the deviating behavior before the analysis to select the meaningful key points that describe this deviation. Improper key points entail a negative bias on a subsequent analysis. Moreover, these techniques still require tedious manual annotation for training, as we can see in this analysis. Here, we have evaluated supervised deep lab cut on estimating red paw posture by annotating different numbers of randomly drawn frames. As you can see, there's an improvement as more training samples and thousands of annotation clicks are harvested. However, there is still a significant difference to an unsupervised approach, which you see here on the left, that learns to compare postures with no training annotations. So to summarize, our goal is to automatically learn to compare postures directly using the pixel-based videos without requiring a detour through key points. We train without requiring body annotations and want to magnify subtle behavior deviations. There are two key steps towards solving this problem that we are now taking a look at. So the first is self-supervision. That is, we learn a behavior representation without supervision from unlabeled data. The key idea is here to establish a surrogate task. So rather than relying on posture annotations, we have our learning algorithm solve a temporal ordering task. That is, we predict the temporal order of a video sequence. To solve this jigsaw, the AI needs to understand the causal temporal ordering of behavior. Since our proposed machine learning algorithm would go beyond the scope of this venue, I have to refer you to our ECCV publication here and the journal reference listed above. Second, we want to support a direct comparison of behavior across subjects. We want to magnify even subtle differences from reference so that they become directly visible. But how can you find and magnify subtle differences when overall posture and appearance drastically changes between different subjects. In a microscope, typically everything is fixed, but the one thing that you want to magnify. And in our case, all other influences, such as overall body movement or appearance variations, are much larger than the small deviations that we want to magnify. Therefore, we learn to disentangle the subtle posture deviations that we want to magnify from other factors, such as appearance variations. Our goal is to train posture encoding so that it allows a linear extrapolation in this learned feature space between query and reference posture. That is, we magnify the characteristic deviations in posture between query and reference. And then we can take this magnified posture and recombine it with the query appearance to render the subject in the query with its characteristic deviation from the reference being magnified, but the appearance taken from the query, so that it's directly comparable with the original query frame. So let's evaluate this disentangling. Can we take these subjects in the columns here and render them with the posture of these queries here in the rows? Let's see. Yes, indeed, we can see that appearance is correctly synthesized along the columns. You see that this remains here the appearance remains fairly static, and that the posture is taken correctly from the rows. So they all move here in sync along the different rows. And here you can see the magnification of the posture deviation between these two frames, the reference and the query, for different magnification strength lambda. And you can see how our approach eventually, as lambda gets larger, nicely magnifies this difference in stance, that the stance is actually much wider of the query as opposed to the reference. Here you can see excerpts from two red paw grasping sequences. The first row is the healthy reference, already resynthesized with the appearance of the impaired query. Actually, these had a different appearance. We have now already resynthesized so that you can better compare them. The third row is the magnification of their deviation, which makes the difference in paw rotation in the second column here, as you can see here, much easier visible than if you were to do it directly between the frames. And we can also automatically measure the differences here in the second last row between the reference and the impaired frames. But wait, don't we now magnify arbitrary differences between query and reference? Can we trust that our algorithm only magnifies characteristic differences from a reference and not arbitrary spurious variations? Here you see the deviations of impaired sequences from a healthy reference. And you can see the corresponding differences here highlighted in gray. And here are the deviations of healthy from the healthy reference, different subjects utilized in that case. And you can see that the magnification does not show any differences. And therefore, there is no arbitrary magnification happening. In that case, if the behavior of your query is very much related to your reference behavior, the algorithm doesn't apply spurious magnifications. Here we see the behavior representation is detailed enough to capture even the differences between a mouse post-stroke and a healthy reference behavior. So directly visualizing in a TSNE representation here in this feature space, the differences in behavior. And on the right-hand side, you can see the projection of our representation being suited for differentiating between different neurodegenerative diseases in human patients. But can we go deeper? Here we have trained rats for a month to grasp. And we then compare their behavior as training progresses against that of trained rats here at 0 days. And you cannot only see that their behavior gets ever closer to the trained state, but you can also see how it is getting closer. We can see prototypical postures that are early on here when training starts, underrepresented in blue or overrepresented. So we can see the postures which are lacking, which haven't been, the rats prototypically don't exhibit, or here in blue, or represent too much compared to trained animals. As a last experiment, let's see whether there are correlations between particular behavior and changes thereof with individual cortical areas being active or inactive. That is, we optogenetically switch individual circuits in the brain and automatically measure the deviations that result in behavior. So is there particular behavior triggered by individual circuits being on or off? Let's see. So is the behavior representation essentially sufficiently detailed to recognize optogenetic switching of cortical circuits? Here we take animals with optogenetic silencer, the red cohort, which you see here, and a control group that does not have this silencer, so which essentially does not react to the optogenetic switching. And we try to classify based on the behavior if the laser was on or off at a given point in time. Now for control, we obtain chance level performance as was to be expected. And for the reference, a significantly better accuracy. Note that we would not expect 100% since optogenetic switching only impacts specific motor behavior and not all individual postures. And so the difference is only visible in parts of the grasping sequence. Our tool is readily accessible via web interface, and the code is on GitHub with the link above here. So in summary, uBAM automatically learns to discover, to quantify, and to compare even subtle deviations in motor impairment across subjects. It provides an easily accessible direct visualization of differences directly in the video. And the approach is based on a self-supervised strategy for disentangling behavior and posture that does not require posture annotations, nor does it require a detour via keypoints, which would imply a strong inductive bias. Thank you for your attention, and thanks to my wonderful group and our collaboration partners on this project that made this work possible.